Thank you very much, and also thank you for having me speak here. So I'm going to switch gears a little bit and talking about, instead of microbes, I'm going to talk a little bit more about chemistry in the human habitat. And so one of the things, outcomes of the Human Microbiome project was really fantastic. Is basically you have a great parts list of the microbes that live in and on us. And so that's a pretty major advance, because then you can start to have discussions on what they're doing. The second aspect of this is now we're starting to become better and better at also talking about the chemistry associated with a person. But we spend 90% of our time inside. And so if you ask the question, what are the top 10 most abundant molecules that are present within a human habitat, everybody will stare at you with a blank face. And this is pretty much universal. And so if we cannot answer that question, how can we begin to study the interaction between the chemistries, the microbes, and the habitats? And so if you want to begin to develop theories, you need to be able to take an inventory of the parts list, so to speak, in the same way that Darwin did it for finches, that ultimately led to the theory of evolution. So if you want to be able to take control of the microbiome in a human habitat, we need to be able to see and understand what the parts are. We can think about a little bit of where these parts are coming from, at least from a chemistry standpoint. They come from building materials, they come from indoor air, they come from surface reaction, and this is really where quite a few people have spent some time doing research on, are these three areas. But what we have not done very well yet is to really understand how the occupants impact the chemistry of the human habitat. And so, simple questions, how much do they contribute to the chemistry in the indoor environment? What type of chemistry, when, and where do they contribute these different chemistries? So let me start off with the first question, is what do people contribute? Well, I can't answer that directly, but I can maybe indirectly answer that based on this project that just got accepted. This is a forensic study that we did. Here, the basic idea was if you leave behind an object, you leave your phone behind or you lose your keys, and let's say this happens at a crime scene and an investigator wants to figure out who that key belongs to, and if there are no fingerprints on those keys, you have no way of figuring out that particular individual. And so the idea that we had was, well, what if we just look at all the chemistries that are there? Can we create a composite sketch based on the kind of medications, kind of food, body care products that we see on those objects? Can we begin to sort of classify the group of people that those objects belong to? And that would help an investigator. And so what we did in this particular study, we took 39 people, we sampled their phones as well as their hands, and then subjected these to metabolomics analysis. And so when you look at this from a 30,000 mile view, you can actually ask the question is, can we actually correlate the hands of those particular individuals to the phones to whom they belong to? And indeed, in 90% of the cases, we were able to even get a perfect match rate. But our goal was not necessarily to identify the person, but what this really shows, we wanted to build a lifestyle sketch, but what this really shows is that the majority of the chemistry must be the same between these between the hands as well as the phones, and that means that people are transferring molecules, and perhaps the objects themselves are transferring molecules to the people. And so a hint of the type of chemistries that might be observed on these particular objects, and that could be an environment, could be a chair that's sitting here or your own chair that you're leaning on. What type of chemistries are transferred to the human habitat in many ways? And so what most people don't tell you when they talk about metabolomics, how difficult the annotation process is, particularly if you do untargeted metabolomics. And so typically what we do in mass spectrometry, we detect molecules, we weigh molecules, but then what do we do with it? Well, because the community knowledge that exists to annotate the chemistry from metabolomics data is poor at best, we built this crowdsourced metabolomics analysis platform with Nina Badera and computer science at UCSD. And what we do here is we basically take untargeted metabolomics. So think of it this way. You collect, you weigh molecules with a mass spectrometer. And typically in an untargeted metabolomics experiment, in order to try to get an annotation, we isolate those ions inside the gas phase. And then we hit it over the hedge with a sledgehammer. In our case, our sledgehammer is helium gas. And the chemistry dictates how these molecules fall apart. And so that creates a barcode signature for that particular molecule. And so we have a whole series of these barcodes, as you will. But that also means if you have two molecules that are very similar to each other, that these barcodes are going to look very similar. And we can use that to organize the information that we can collect by mass spectrometry. And so we align the spectra, and then we get these aligned spectra. And these are then we think of these as aligned barcodes that represent groups of molecules that are related to each other. And then we represent them, instead of spectra, we represent these as nodes. And so now we get this group of molecules that are often separated, let's say, by an oxidation state, or if you have looking at fatty acids, some different methylation patterns, et cetera, et cetera. So that's how they group together. So how do we take advantage of this when we looked at it into our hands-on study? Well, we created one super molecular network. This is what we refer to it as. There's no biology necessarily. But then you can add sample information to it. So you can color-code it based on the molecules that came from the volunteers. You can color-code it based on the molecules that came from the phone or that are shared between the two of them. And then, of course, any annotation that is in the public domain where you have reference data of known molecules, they are automatically mapped as well. And so in one picture, you get all the chemical information that you can collect in a given untargeted metabolomics experiment. So when we go to our hands-on study, it turns out that 53% of all the molecules that we were able to capture by mass spectrometry in this particular study were shared between the hands and the phones. That's how much transfer takes place. And I imagine as I'm sitting on the chair that similar numbers take place as well. So here just a very small selection of molecules that we see. We found the mosquito lotion DEET. We found the antiseptic chlorhexidine. We found sunscreen, avobenzone. And so these are clearly human-derived molecules that are then transferred to an object, basically. And so in our case, we can actually use this to learn sort of the lifestyles of those particular individuals that own those particular objects. So here we know that this person is a vegetarian. They like to go camping, spend a lot of time outside in the sun, based on the amount of sunscreen that we see. And we can go through this, including all the medications, the type of foods that those individuals eat. So we can often tell an Indian person versus an American person or a Mexican based on the type of diet and type of molecules that we see in these lifestyle sketches. So that was only the phone. But the question is, well, where are some of these molecules? Where do they end up inside the human habitat? And I'm going to just highlight a class project that just got just got published. This was a project where I was teaching the class on how to do mass spectrometry and how to interpret mass spectrometry data. And we started to think about, okay, what would be a sample type where we know very little about? And the human habitat is one of those sample types where we actually know remarkably little about. And so we went through 3 different human habitats, so an apartment, a bar type setting, and a water fountain area. And then 2 modes of transports, bikes and cars. And so when we take our inventory of our molecules, so to speak, you can just simply color code them. You can color code the network. And very quickly, you can already see there are unique molecules that are only found associated with the cars. There are unique molecules that's only associated with the happy hour, et cetera, et cetera. So there's really a unique chemical distribution. I really wish I had that access to that 1,200 study sample from the 1,200 homes. To see if this is a general trend where there's unique chemistry in each one of those homes. But when we start to look at the frequency of these molecules, there's some interesting molecules that actually originate from human usage. So here we have propylbetaine. Those are a common ingredient to shampoos and other types of soaps. And you can see in pretty much nearly every single sample we investigate, we find this particular residue. Caffeine, it's in about, 80 of the samples that we looked at. And then even erythromycin, which is an antibiotic that's used by people, you can see that that's in about 12 or 13 samples. So even medications that we take end up in the human environment. And I won't talk about cocaine, but that ended up, we found that on a football table. But we can even begin to look and map this in 3 dimensions, and I'll go into it in a little bit of how we create these kind of 3-dimensional maps. But here you can clearly see molecules. So this is a car that belonged to person 1, and you can clearly see that the molecules that are on his shoes ended up on the bottom of where the gas pedal is. So these are the kind of relationships. I don't know if it's a molecule from the gas pedal that's being transferred to his shoes or vice versa, but there's clearly a relationship between the person and the human habitat. Same thing with caffeine on the bike. You can see that caffeine, person 1 drinks a lot of caffeine. You can see it nicely on his hands, but you can also see it on his helmet as well as his bike handles as well as his bike seat. Whereas person 2, who doesn't use either of the bikes, they don't drink any caffeine, and then 3 and 4 do drink caffeine. But for some reason that didn't show up on that bike. But that person is very clean. He washes his bike probably once a week, which is pretty remarkable. But we can actually begin to trace chemicals throughout the entire apartment. So this is a apartment that belonged to person 1 and 2. And you can see that person 2 has a little bit less propylbetaine. Most of her personal care products don't have that particular detergent in it, whereas person 1 uses a lot of cocamidobetaine containing detergents. And so what you will see is that room 1 belongs to person 1. You can see a lot more of this detergent there. And in fact, you can even tell that this person sleeps on the if you're looking this way, the right side of the bed as opposed to the left side of the bed. And a little bit less of this detergent is found in room 2, but you can still see traces of it even though that person uses less of it. And so if you start to think about what contributes to the chemistry of the human habitat, the occupants really provide molecules that originate from cleaning tools, from personal care, including medications that they use, the food, drink that they have. We even have evidence of the clothing that they have. So these are some of the chemistries that we see. But how does this chemistry relate to the microbes that are there? Well, that gets us to the office study that Rob introduced a little bit earlier on. This is my office. It's a mess, I know, but if you look at where we sampled, those are all the yellow locations is where we collected a sample for both 16S analysis as well as metabolomics analysis. And after we obtained the metabolomics data and mass spec data, what you get is a table that looks like this. You get a table that where we have XYZ coordinates. Then if you look at F, G, and H, you can see the bacterial feature, or it would be a metabolomic feature, and then below that we have an intensity map. And we have literally millions of these things. And then we need to make sense out of them. And that's where we map that data back onto these 3-dimensional images so we can begin to interpret it. And as Rob said, yeah, that's a very expensive way to figure out that somebody spilled caffeine on my floor. But on the other hand, without visualizing, it would have been really hard for me to determine that somebody did spill caffeine on my floor. But you can begin to trace back a lot of other things, and this also illustrates how long some of these chemistries really last. So this is DEET. This was a part of a trip. I went to Mount Hood. I went backpacking on Mount Hood in Oregon. And this was 6 months before we did this sampling. And you can clearly see my phone lights up still with DEET. And even my desk lights up with DEET. And so some of these chemistry really last a long time, and we have no knowledge of how long chemistries really last after people have transferred some of these chemistries in. we can look at the pKa data of the metabolomics data, and you can start to think about the relationship of all the other samples to the floor samples. And yeah, you can try to interpret it, but in the end there are so many different ecological niches that exist within one office alone that is really hard to interpret. But if we translate this data, and particularly the relative intensity of the samples from the floor to all the other samples, now it becomes interpretable. Because if you look at the blue data, that's the floor data, and the further away you are from your floor based on the pKa space, meaning that the chemistry, the overall chemistry starts to look very different as you move away from the floor, you can nicely see that in these particular images. And so we can even zoom in a little bit and actually start to begin to interpret some of this information. So if we look at the floor, you can look at the bottom of my foot even, and you can start to see that the bottom of my foot is most similar to the floor. That's the area that's most similar to the floor, and it makes sense that that's the case. But as you go up my leg, basically, you can start to see that the chemistry becomes more and more divergent from the floor chemistry. The same is true for the microbes. Again, if you look at the microbes on the floor compared to the bottom of the feet, they're very similar. But then if you go up your ankles all the way to your knee, it becomes more and more divergent from the floor microbial communities that are present in this particular office. And the question is, does this hold true for many other office environments? Here's just a different angle of the microbial data. And again, you can look at these are Rob's legs. You can see that the bottom is blue, and so that basically shows that the microbial community is most similar to the floor microbial community. And so we can even look at individual microbes. So here we have Prevotella. Here we have Stringomonas. Sorry, that went fast a bit, but Prevotella is basically associated with one particular individual and any object that that particular individual has touched. Sphingomonas, which is a soil microbe. I'm pretty certain that Sphingomonas is not growing on my carpet, or I assume not, but it's probably because people have tracked those in. And then you can see Synechococcus. It's all over me. You can see it all over my clothes. I do spend some time in the ocean, and so maybe that's the reason why you can find it there. Do the chemical niches really matter for the microbiome in the human habitat? We only said, okay, the microbes are tracked in by people so far, but are there chemical niches that matter? Yes and no. I think yes, the data suggests yes, because if you create a pKa plot of the metabolomic data and then color code it based on the organisms that are most divergent for each sample location, you can see that they group with specific chemical environments. And so that suggests that some microbes care about the kind of microbial environment they're on, whether they're in carpet, wood, or on the ceiling material. But no, it's because humans really track in a lot of chemistry and a lot of the microbes, and they dominate quite significantly. So here, the Actinomycin nocardiae, it's nicely on the floor. This is again a soil microbe, and I would imagine people who are responsible for tracking it. In fact, we can take advantage of this if we ask the question about person 3. So take all the chemistry that person 3 has on their body, which is me, and ask the question is, what is the most likely place that this person has touched? You can see that it lights up very nicely throughout the entire office. My desk, my table that's sitting there, even the floor. Apparently I don't touch my wall very often, and occasionally, but rarely, I touch my bookshelf. But you can do this for person 1, person 2, person 4. You can do the same thing for the microbes. And again, if you look at person 3, you can see my microbes are all over my objects. Rob's microbes, this person 2, you can see his microbes as well as his chemistry are all over his objects, his mouse, his computer, etc. But we can create these sort of independent images for every single molecule, every single microbe that we see. And so we need data tools that allow us to be able to manage this type of information. Finally, I want to leave you with some take-home questions, basically. Some of the fundamental questions that I think are interesting from a human habitat standpoint is what are the most important processes to sustain microbial life in the human habitat? Are there microbe interaction, microbe surface interactions that are important that could define a healthy human habitat? And can we take control? I think that's ultimately the question. Can we charge engineers and architects of how we can take control of the microbiome? And then finally, I need to thank some people. This is a large list. These are the people who, this is the GMPS community that really facilitate the building of this crowdsource molecular analysis platform. And then, of course, friends, co-workers, and that really supported it. Nuno Badair for molecular networking, Theodor Alexandrov for the 3D, and then the Night Lab for sequencing, and then Sloan Foundation particularly for funding some of the work on the indoor human habitat that I showed you. Thank you.
So I think the plan is to have both speakers come back down here and have a joint question and answer session.